Our next speaker, who is Dr. Nicholas Douville, who is a clinical lecturer in the Department of Anesthesiology at the University of Michigan. He is going to share with us some lessons learned while using the Michigan Genomics Initiative data in the operating room. Again, if you have any questions for either Dr. Ganesh or Dr. Douville, please submit them using the Q&A tab. And then after Dr. Douville is done, Dr. Ganesh will be coming back and will be answering those questions. Thank you. You are muted, Dr. Douville.
You can hear me now? Can you hear me now? Perfect. Thank you for the kind introduction. My name is Nicholas Douville. I'm faculty in the Department of Anesthesiology and Critical Care Medicine. And for the past 2 years, I've been using precision health data and Michigan Genomics Initiative data to study how genetics can impact my everyday work, which is caring for patients in the operating room and the intensive care unit. I have no conflicts of interest to disclose, and we don't discuss any off-label uses of medications in today's talk. Since I don't...
Sorry, I was muted. I do have one question. Do you have slides?
Oh, are they not sharing?
They are not showing.
Can you see them now?
Yes, we can.
Perfect. You're seeing them now? Good. Since I'm going to spend the rest of the presentation talking about complications from surgery and how our research seeks to minimize those risks, I thought I should take one slide just to reassure the entire audience that a trip to the operating room is very safe. In fact, the overall population mortality is about 1.1 per million patient years. And perhaps more importantly in the context of today's talk is that as anesthesiologists and clinician scientists, we've sought continual improvement and continuing to become safer through time with an exponential decrease in both the rate of complications and the rate of mortality over the last 70 or so years. I wanted to motivate the talk. So why is the perioperative period or the time surrounding surgery, why is that an appealing target or opportunity to use precision medicine? The first reason is that vast quantities of data are generated with each trip to the operating room and the surrounding postoperative period. This can include a variety of factors: patient demographic information, family history of different disease processes, documentation of social issues like tobacco, alcohol, or other substance use. It also, as you can see in this picture here, when I'm taking care of a patient in the operating room, a large amount of vital sign data is being generated in terms of their blood pressure, their heart rate, what their oxygenation level is. Large amounts of medications are administered, and those are recorded with high fidelity in the perioperative period. Billing and diagnostic codes are generated, and there's a lot of intraoperative procedural details, such as the length of the case, what fluids are administered, and if any blood products or transfusion happens. Another reason is that a large percentage of complications that do occur surrounding the time of surgery. This study from Dr. Dimmick in the Department of General Surgery shows that approximately 13% of patients will have some complications surrounding the time of surgery, and that about half of those will be classified as a major complication. And that includes things like pneumonia, stroke, unplanned or inability to come off of mechanical ventilation. And the two that I wanted to specifically talk about, because we'll focus on them later in the talk, are myocardial infarction after surgery and clotting or thromboembolic complications. Final reason why precision medicine makes such an ideal, or the perioperative period is such an appealing target for precision medicine, is that we spend a disproportionate amount of the overall healthcare budget surrounding the time of surgery. This vast amount of data that's generated around the time of surgery now increasingly includes genetic data. This might be from sources like direct-to-consumer genetic testing, genetic data that's available because it was obtained for unrelated causes like cancer workup. And then now at Michigan Medicine, we have genomic data on over 70,000 patients that have enrolled in the Michigan Genomics Initiative. Despite this widespread growth, healthcare providers are given limited exposure to genetic data and are not really trained to contextualize or clinically act upon this information. So the overall objective of my research is to perform perioperative genomics in order to assist healthcare providers in improving patient outcomes through a unified platform that identifies patient attributes that may affect their care, which we call these comorbidities, and stratifies the risk of key perioperative complications. So the overview of today's talk is really going to focus on the two aspects: the comorbidities, which we've studied, and the initial work have focused on three of high interest to anesthesia in the perioperative period: malignant hyperthermia, pseudocholinesterase deficiency, and factor V Leiden. And really one perioperative complication, myocardial injury after non-cardiac surgery, which we'll call MINS through the rest of the talk. Despite only focusing on the one complication, the overall goal is to really stratify and look at the risk of a variety of complications, and a few of these that could be studied for future work are sepsis, pulmonary complications, stroke, and kidney injury. So first, what made us select the initial comorbidities with which we studied? These should have had or were selected because of straightforward genetic inheritance, a modifiable risk profile, a penetrance linked to anesthetic exposure so that they would have a potential misdiagnosis using traditional techniques, and high clinical significance. The three that we ultimately decided to study for our initial work were malignant hyperthermia, which is a life-threatening hypercatabolic state following anesthetic exposure, which leads to fast heart rate, muscle rigidity, fever, and muscle breakdown. The genetics of malignant hyperthermia are a mutation in the proteins responsible for calcium regulation in skeletal muscle, this RYR1 and the CACNA1S genes, and that they should be modifiable. And specifically for malignant hyperthermia, providers can avoid anesthetic exposure to volatile anesthetics and succinylcholine, which can trigger a malignant hyperthermic reaction. We also selected pseudocholinesterase deficiency, which is a deficiency in an enzyme responsible for the metabolization of choline esters, such as succinylcholine and other local anesthetics. The population that's heterozygous will have a slightly prolonged response to these medications, but really clinically significant is the homozygous population, which can have a profound response with paralysis lasting anywhere from 2 to 8 hours. Pseudocholinesterase deficiency results from a mutation in the BCHE gene on chromosome 3, and providers can again modify their anesthetic plan by avoiding pharmacologic agents. This can avoid or minimize potential complications like a delayed emergence, a prolonged requirement for mechanical ventilation, ICU admission, or local anesthetic toxicity. The final comorbidity we selected was factor V Leiden, which is a mutation in the clotting cascade, which predisposes individuals to blood clot formation. It's caused by a mutation in the factor V gene or coagulation factor, and it's modifiable because the providers may work together to select a different choice of intraoperative anticoagulation, which can lower the rate of prothrombotic or clot formation. So the hypothesis for the first study on these comorbidities was that patients who may be at increased risk for modifiable complications can be identified using genetic data obtained from their enrollment in MGI, and that such patients may have previously missed diagnoses on any of the traditional EHR-based documentation. So key steps on this project: we developed 3 EHR-based, so these are non-genetic phenotypes that were created from a combination of ICD billing diagnoses, and then free text from the preoperative history and physical. We assessed the ability to detect these genetic changes, and notably malignant hyperthermia had inadequate coverage in the GWAS data, so we had to then pursue a subset of MGI patients who had additional genetic data available that we could assess for malignant hyperthermia. We also did a confirmation of the genotype to phenotype linkage, and that was through the genetics of ABO blood type with the EHR phenotype, with which these patients often have blood type laboratory studies obtained as part of their preoperative workup. And then we performed a limited clinician adjudication to confirm the phenotype fidelity. And then we also assessed and did a chart review of the positive cases. So a quick summary of the results. We had over 40,000 patients. This was data that was froze from, I guess, almost a year ago. So we only had 40,000 instead of the 70,000 that are now available. And out of those 40,000 patients, we had 25 patients who were homozygous for pseudocholinesterase deficiency. And of those 25, 56% had a pre-existing diagnosis. And then we also had about 5% of the population was heterozygous for pseudocholinesterase deficiency. For factor V Leiden, we had 29 patients that were homozygous, and of those, 3 were known about from the EHR automated phenotype. And then on a clinician review, I found an additional 3, up to 21%, that had evidence of factor V Leiden somewhere in their chart that was missed by the EHR phenotype. And then again, about 5% were heterozygous for that condition. As we said, malignant hyperthermia, we were only able to assess that in a subset of patients. We had those additional data forms in about 1,000 patients. 2 patients were identified with an associated mutation, and neither of those patients had a previous clinical diagnosis of susceptibility. So some conclusions. We identified patients with genetic susceptibility to complications using an open source script, which we designed for clinician use. We validated this in a retrospective analysis of 3 conditions with well-characterized inheritance. And we showed that not all genetic susceptibilities are previously known or documented in the electronic health record. So then moving on to the second part of the work we did on perioperative complications, we selected myocardial injury after non-cardiac surgery. That was selected because it's of high clinical significance, relatively high frequency, and there was an objective troponin-based diagnostic metric, so we could easily refine and define the phenotype without having to do a lot of complex post hoc analyses. To put our work into perspective, I wanted to take the next couple slides just to describe how perioperative complication risk is currently being assessed, and that'll put into perspective what we do unique and differently in our project. So the first and most widely utilized risk assessment for cardiac complications is the Revised Cardiac Risk Index, or the RCRI. Basically, it's a 6-point scale where a provider can assess, do they have ischemic heart disease? What's the risk of the surgery? Do they have congestive heart failure, et cetera? And then based on the number of points they have on this metric, their risk ranges from less than 1% if they have 0 or 1 risks, all the way to greater than 11% if they have 3 or more points on the scale. So advantages, the reason it's widely used is that it's very easy to provide a kind of back of the envelope calculation of an individual patient's risk. But the real drawback to this is that it's not really physiologic. Why should a patient get the same risk assessment if they have had a stroke or if they have diabetes requiring insulin? So it's really oversimplified to allow widespread use. A more complex but probably less utilized risk assessment metric is from the American College of Surgeons. And either a healthcare provider or a patient can input a variety of answers to a variety of questions. What type of procedure are they having? How old are they? Age? What type of history do they have? Are they on medications for high blood pressure? Have they ever been on dialysis? What's their height and weight? And then it outputs the risk of a variety of complications, including heart conditions, but also stroke, delayed removal of the delayed requirement for mechanical ventilation. The major drawback, which has impacted its utilization, is that it's not integrated with the electronic health record. And so it really requires someone to manually input this, and that's not something that providers are really equipped with the time to do. Another impact specific to what we do is that there's no genetic information incorporated to the risk score, despite evidence that may explain a significant portion of the overall risk. And finally, as many of you are aware, there's been significant improvement in the ability to assign polygenic risk for a variety of conditions that has occurred over the past few years. However, currently, these are limited from our perspective because there's not any perioperative phenotypes. Nobody's specifically looking at the risk of complications around the time of surgery, and we know there are unique stresses that occur with a trip to the operating room. And also they tend to focus on the isolated genetic contribution without integration from a variety of other clinical data sources which are available. So the hypothesis for this portion of the project was that the addition of polygenic risk scoring can improve our ability to predict patients that are going to develop MINS. and that a more diverse set of preoperative and intraoperative variables would lead to improved MINS modeling compared with the benchmark which we used, which was the RCRI cardiac index that we talked about a few slides ago. So key steps on this project were, first, we needed to replicate polygenic risk score, and we did that with the polygenic risk for coronary artery disease that had been previously described in the literature. We replicated that in Michigan Genomics Initiative data. Then we also worked to generate new polygenic risk score for a novel or a new phenotype, which was specifically the myocardial injury after non-cardiac surgery. And some key lessons that we learned is that there are differences in the ability to define any given phenotype based on data availability from each data source. The way that coronary artery disease can be defined, or MINS can be defined from UK Biobank, as somewhat different from what we can define in MGI, specifically because we have access to more rich data on the intraoperative course, laboratory studies such as troponin, so we can really define the phenotypes in higher fidelity, but perhaps not identically to how they're done in traditional sources like UK Biobank. Then also really the necessity of independent validation sets that can then replicate those definitions we do was a step that or an issue that we encountered. So you can see in these three figures that we have here, it's just coronary artery disease in Michigan Genomics Initiative population, myocardial injury from UK Biobank, and then myocardial injury from Michigan Genomics. And so there's differences in both the overall incidence and rate, and that's based on the definition and population from each of those groups. So a quick summary of our overall results. We had data on 90,000 unique procedures on over 40,000 patients with over 400 cases of MINS for a rate of 5%. Polygenic risk was independently associated with MINS in each of our models. The RCRI model, a model censored with just the data available at the time of going to the operating room, and that was the preoperative, what we call the preoperative model, and then a model that incorporated both preoperative and intraoperative data that included all the data we could get from a trip to the operating room. And really each risk factor improved model discrimination. So our pre-op plus intraoperative model was better than the pre-op model, which was better than the overall simplified model from the RCRI. And notably, polygenic risk score did not improve discrimination, but a larger percentage of patients, up to over 3.6%, were shifted to a new risk classification when you included the polygenic risk data. So we're presenting here a population density distribution. You can see statistically significant differences in both the mean, in population mean between those patients that did not develop MINS. Their mean was 0.01, and that's on a standardized polygenic risk score, so mean normalized to 0 with a standard deviation of 1. Then those that did develop MINS had a mean that was higher at 0.14. And then if you look at what the individual risk is a function of polygenic risk score, those that had a polygenic risk greater than 1 standard deviation had a 1.36-fold increase of developing MINS, and those that had 2 standard deviation greater than the mean of polygenic risk score had a 2.1. And then just another way to look at this data, this slide here is showing the -1 to 1 is in blue, so that's patients with what we call a baseline polygenic risk at each of the 4 RCRI points, so those at RCI 0 risk factors all the way up to patients with 4 RCRI risk factors. And so you can see exactly what you'd expect in the baseline risk, that as you have more of these traditional risk factors, your overall risk of developing a myocardial event after surgery increases. But what was notable was that at lower risks, specifically 0 to 1, genetics had a relatively small contribution. So even if you were very high genetic risk, it didn't really shift you into a higher overall rate. Whereas at high overall risk, 3 or 4, genetics had a huge influence. And you can see that the population that had a high genetic risk actually accounted for over an RCRI point, and those that were low basically subtracted an RCRI point. So genetics had an oversized or a larger contribution than the non-genetic factors at high overall risk. So some quick conclusions. Genetic information associated with MINS is useful for identifying patients who are going to develop a complication after surgery. The diverse variables improve modeling, with pre-op variables better than traditional models, and pre-op plus intraoperative variables, things like case duration, blood product transfusion, requirement of certain medications could increase the risk even more. The integration of genetic data improves predictive value compared to either genetic analysis alone or the RCRI model. So next steps for our project, we're currently navigating the ethical and logistical steps to return the genetic findings. And that's something, as a clinician scientist, very meaningful to see how the data we obtain in MGI and the work we're doing will then go back when these patients subsequently have surgery, how it will impact their care. And we're working as a first line to return the genetic information to the 2 patients we discovered that had a malignant hyperthermia risk, so that when they undergo surgery, the provider will directly modify their care. We're expanding the list of perioperative complications, and that's what I showed at the start. Things like stroke, kidney injury, delayed emergence from mechanical ventilation. We're working to automate the extraction of variables with the electronic health record. And we're working to integrate some of these risk scores in a clinical decision support. This is something that was developed by Dr. Tremper in the Department of Anesthesiology for non-risk scores, but it just provides the provider a clinical decision support. You can see any of the areas where you're potentially could make a error or oversight in your anesthetic management, and you get alerts in an easy to read way about each of the kind of core systems that you'd be monitoring, from the brain to the heart to the lungs. We envision something analogous for overall risk scores that a provider could, they said, get the information they need that incorporates genetics into a scoring system where key complications or comorbidities are flagged in a way that integrates with their regular workflow. These are some mock-ups of how that might occur. And really the overall lesson from the last 2 years that I've been using Michigan Genomics Initiative data is that the success of the project has ultimately hinged on the collaboration and support of really the unique environment we have at University of Michigan. And again, learning the genetics side, I've received tremendous support, mentorship from the entire Willer lab, a lot of other support from the Precision Health Office, Data Office, and Michigan Genomics Initiative, and then my own department in the Department of Anesthesiology. So thank you.
Great, thank you so much, Dr. Douville. So it looks like we have just a limited amount of time for questions, but we do have a few questions. One of the questions is we'd love to hear from both speakers how they envision putting the information into the hands of clinicians who aren't precision health experts and promote awareness of such tools. Dr. Ganesh, are you able to join us? Yes, thank you. Dr. Douville, do you want to talk to this since you are currently in that kind of conundrum of not being able to necessarily share results because of regulations, but wanting to do so, and how you would envision doing that?
And that's exactly a question that I would love to answer. We have the 2 thrusts. One is the immediate. It's just if you find an actionable genetic piece of information that you'd like to return, such as malignant hyperthermia, that seemed that patients would almost universally want to know that, because it can lead to directly modifiable changes in the way their care will be provided. So we've worked with the IRB, worked with Michigan Genomics Initiative, and are in the process of returning that patient data to them. Ultimately, we're still defining what the best practices, and we're learning on that, but it involves doing additional CLIA grade clinical testing for them and providing them access to genetic counseling so that they can then integrate that from a research grade finding into a clinical grade finding. So we're navigating all of those issues. I think we have, hopefully the next time that we meet or that we talk, we'll have lots of success stories with that, because that's something we're ultimately pursuing and are in the process of doing, and I think we have a good path forward. Then the integration with healthcare providers, long-term goal is, as you could see from some of the other risk metrics, very difficult to incorporate that seamlessly. So it would be integrating whatever genetic data is available with automated factors in a way that healthcare providers that may have limited training in genetics and have limited time available to devote to each patient can really get that. What is the take-home message is that this patient has a 3-time greater than baseline risk of developing a heart attack after surgery, or something about this patient's genetic means they're likely to have a slow wake up from anesthetic. Maybe you should avoid drug A, B, and C. And so that's something that the implementation will really be what determines the success or failure of the ability to do that within the constraints of what healthcare providers' time and training allow.
I think too, later this afternoon, we're going to be hearing from the Precision Health Implementation Group, and they are thinking very deeply about all the issues that you have brought up, as well as how do we take this information and put it into the hands of people that can use it clinically, so the non-experts, so they can understand it. Let me see, there's another question in here. Okay. There's a question from Dr. Ullman. I'm assuming we're happy to see the 2 participants in our medical genetics clinic to offer CLIA testing for malignant hyperthermia. So you could send them there as well into the medical genetics clinic. So that is it for our questions. Are there any last comments from either of our panelists to the audience before we break and go to the poster session?
No, I'll just share that we're doing something very similar, but our cases are coming from outside of MGI, so we have a process for returning results that is engaging with genetic counseling as well. And for the actionable finding we talked about, for example, getting that CLIA certified testing and then returning it in the context with counseling is really essential.
And for those of our audience members who may not understand what CLIA certified testing is, it is a laboratory that has all the necessary approvals to do the testing, making sure that you get your test results back, and that's part of the process in place. So laboratories lab values that you get done in the hospital go through the CLIA certified process. So it's kind of this nuance between research and clinical practice, and wanting to make sure we have the best information available. So thank you both for those really amazing talks. It's so great to see MGI being used as a resource for improving care, as well as learning from you about others that might be interested in this, what missteps or issues you faced, so that we can improve the community of users around MGI. At this point, we are going to be taking a 10-minute break. The next thing that is on our schedule, as you can see from the slide that's on the screen, are our poster sessions. So this is something new. And if you've not been part of a virtual poster presentation session before, please do join in. We have 28 poster presentations, many of which are being presented by some of the trainees or students that are doing precision health research here at the University of Michigan. You should have received a link or in an email that tells you where to go. It is a separate system from this Zoom system. You don't have to leave this Zoom system. You can open up the other link. The two are not, they're not connected, so you can be in both of them at once. You can also leave this Zoom system if you'd like, but please join us back again at noon when we will have additional panelists. To chat with the poster presenters, if you go to the link and take a look at the different topics that are in there for the different posters, you then click on the chat button, and that will put you into the Zoom room with that poster presenter. And you can hear about their science, ask them questions, and interact like you would, although it's probably going to be a lot quieter than most abstract and poster halls, which can get very loud with everyone talking at once. So I guess that's one of the benefits of doing this in a Zoom room. You'll see on the screen here, here's an example of what our poster rooms look like. So please feel free to go there and chat with our poster presenters. Again, we'll be back in this space promptly at noon with the afternoon session focusing on how precision data is being used to improve patient care. Great segue from this morning with some real life applications, and it'll be exciting to hear how our investigators are doing that. So the link to go to the virtual poster session has also been put in your chat room. So please go there and after our quick break. Thank you.